So Ritu, what happened? You will explain the structure of the DNA, whatever we have done. Ritu, are you there? Acha, mic not opening. Okay, so can Barbie explain by the time Ritu is trying?
Yes, ma'am.
Okay, so you will start from the beginning. Okay, the whole structure of the DNA जितना अपन ने कर लिया है. हाँ, तो say now.
And DNA is a polymer made up of monomer units called nucleotides.
Very
Now,
good.
the nucleotides are made up of nucleosides plus phosphate group.
Very good.
Now, the nucleoside is made up of deoxyribose sugar plus
Yes,
nitrogenous base.
correct.
Then deoxyribose sugar is a pentose sugar, which is having
हाँ,
5 carbon. It is having the ring-like structure
हाँ,
formed by 4 carbon and 1 oxygen, and the fifth
very,
carbon is outside the ring.
yeah, very good.
Then it is also nucleoside is also made up of nitrogenous base. So
हाँ,
nitrogenous base are of two types:
हाँ,
purine and pyrimidine. So
okay,
purine is having the double ring structure consisting of one benzene ring and
हाँ,
one attached to it is the five-member uh, ima- imida- sorry, ma'am,
imidazole, imidazole.
imida-
हाँ,
imidazole ring.
हाँ, very good.
Purine is having adenine and guanine.
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 Purines are adenine and guanine. Correct.
And then the other type of nitrogenous base is the pyrimidine. It is having the
Pyramid.
single ring structure, that is the member benzene
हाँ,
ring.
okay.
Then pyrimidine is are of two, that is cytosinin and thymine.
Correct. Very good.
Then nitrogenous base shows the complementary um, pairing, that is
Pairing.
the adenine pairs with thymine by double hydrogen bond
हाँ,
and guanine pairs
हाँ,
with cytosine by triple hydrogen bond.
okay. Now, can you tell me the structure of the DNA? ये सारे components तो आपने बता दिए. Okay, now tell me
Okay.
that how these all are arranged in the DNA molecule.
And the DNA consisting of the two strands which are
हाँ,
made up of alternate molecules of uh, deoxyribose sugar and
yes,
phosphate group.
very good.
Then the two strands are joined together by the steps which are
हाँ,
made up of nitrogenous base which are of two types, purine and pyrimidine.
Okay. Then you can say directly the pairing.
That's pairing.
Complementary. Correct. So to which group or which molecule the nitrogenous bases are joined? They are joining the
H,
strand only. So in the strand is made up of alternating molecule of deoxyribose sugar and phosphate group. So these steps are joining to which molecule?
and H and OH.
So phosphate group or the deoxyribose sugar. The steps are joining with the phosphate group or the deoxyribose sugar.
And phosphate group.
No, see in the diagram. Try to see in the diagram. Barbie, you have drawn that other diagram in which the two strands साथ में steps
Yes,
joining
ma'am.
है. तो उसमें देखो किससे अटैच हो रहे हैं प्यूरिन और pyrimidine. एडेनिन किससे join?
Deoxyribose sugar.
You will see there clearly phosphate से steps नहीं जुड़े हैं. The steps, that is the nitrogenous base, they are joining with the deoxyribose sugar. Okay, and then how the deoxyribose sugar and phosphate is bonded? वो अपन ने डिस्कस किया था प्रीवियस पीरियड में, दैट इट इज जॉइंड बाय 5-3 ऊपर अगर 5 है तो नीचे 3 फॉस्फेट से phosphoric acid or the phosphate group is attached. Okay, so that was discussed. And then to the deoxyribose sugar, we find the other one, that is your nitrogenous base will be attached. That nitrogenous base will be either the purine or the pyrimidine. Okay, if one strand is means towards one strand, अगर strand 1 से purine attached है, तो strand 2 के से you will find that the pyrimidine will be attached. So they are showing, since they are showing the complementary pairing, so you will find that one side it will have the purine, so other side it will have the
Pyrimidine.
pyrimidine. ठीक है, तो I hope you are, do you have the figure, that is your 5.6 figure, pairing between thymine, adenine, cytosine and guanine is given. Base pairing. You will have drawn the diagram? No.
Yes, ma'am.
You have that base pairing diagram?
Yes, ma'am. Ma'am, that one
हाँ
you said, na?
हाँ, which I have sent.
Yes,
इसमें
ma'am.
ही भेजा था कि thymine combining with the adenine. Do you have that?
Yes, ma'am.
हाँ, तो थाइमीन में देखो, थाइमीन में नाइट्रोजन दिख रहा है, वो नाइट्रोजन एडेनिन के नाइट्रोजन के साथ में कंबाइन हो रहा है. Could you see? And that is the hydrogen bond. Same way, the oxygen of the thymine is combining with the nitrogen of the adenine. तो इस तरह से अपने दो हाइड्रोजन बॉन्ड से A और T पेयरिंग कर रहे हैं. Did you get that diagram?
Ma'am,
Ma'am,
which is the diagram?
Diagram is 5.6. अगर वो पेजेस के हिसाब से याद करें, otherwise it is written pairing between thymine and adenine and cytosine and guanine. That figure. Just after the nitrogenous bases, the 5 types which you have done. ओके, उसके बाद एक और फिगर आपको भेजा था हमने, जिसपे नंबरिंग है पूरी. 5.8 फिगर है वो.
yes, ma'am.
ओके, 5.8 फिगर है. Two pairs of nucleotides showing bonding between phosphate deoxyribose and nitrogenous bases. तो दिस फिगर everyone has to make. उसमें कार्बन की नंबरिंग भी है. ओके, थाइमीन में कार्बन की नंबरिंग है, एडेनिन में भी है, और किस नंबर के कार्बन के साथ, किस नंबर के नाइट्रोजन के साथ में हाइड्रोजन बॉन्ड्स आर फॉर्मिंग, दैट इज गिवन. You have that diagram or not? Otherwise, I'll send this diagram once again.
Ma'am,
And
5.8
this
is
is,
there, but I can't find the 5.6 diagram.
अच्छा ठीक है, तो 5.8 देखो, 6 की जरूरत नहीं है फिर. 5.8 में everything is there. Okay, 5.8 only is the main diagram where the numbering is given. Neha, you have drawn also in
Yes,
the
ma'am.
notebook?
Yes, ma'am.
हाँ ठीक है. So now try to see. I wanted to explain that it is anti-parallel. The two strands are anti-parallel. Rumisha, do you also have that diagram?
Yes,
5.8
ma'am.
figure. ठीक है, श्रावणी आपके पास है. रितु, यू हैव ड्रॉन. अब आप चैट बॉक्स में ही बोल सकते हैं, अगर माइक काम नहीं करेगा तो रितु, श्रावणी, यू कैन राइट इन द चैट बॉक्स. डू यू हैव दैट 5.8 फिगर? ओके, अच्छा, नॉट ड्रॉन. ओके, श्रावणी हैज ड्रॉन ऑल्सो. ओके, ठीक है, तो इस फिगर को समझने की कोशिश करो. सॉरी, deoxyribose first carbon is joining with the nitrogen which is present at the third place. ये 6 membered ring है. Thymine is a 6 membered ring. सिक्स मेंबर्ड रिंग में थर्ड पोजीशन पर जो नाइट्रोजन है, with that the carbon is bonded. Could you see, Neha, Rumisha, those who have the diagram in front? Could you make it out?
Yes, ma'am.
हाँ, फिर उसके बाद देखो कि थर्ड कार्बन डिऑक्सीराइबोस शुगर का और PO4 which is made, to that you find it is attached. Okay, and yesterday I hope you have taken from the slide how they are bonded. Okay, release of water molecule, by the release of water molecule. So you find, और नीचे वही phosphate is bonded with the next deoxyribose sugar with the fifth carbon. So above it is bonded by the third carbon, but down it is bonded with the fifth carbon. Did you get it?
Yes, ma'am.
हाँ, दोनों स्ट्रैंड समझ में आ गए. अब see your second strand. When you see the second strand, you will find the deoxyribose molecule, it is inverted. Did you get it, children?
Yes, ma'am.
It is inverted. अपन को एक स्ट्रैंड में O ऊपर दिख रहा है, in the other strand the O is upside down. The whole molecule is upside down. तो अब here in this case you find that the third carbon, ऊपर अपन को phosphate दिख रहा है, where you find that the third carbon is bonded with the phosphate and the fifth carbon down it is bonded. So you find it is turning around. So that is why we say that two strands are anti-parallel. Two strands are
Anti-parallel.
anti-parallel. अब if you see in the one strand, तो fifth carbon free है. And if you see the other strand, उसमें अपन को थर्ड कार्बन दिख जाता है फ्री, जिससे आपको फॉस्फेट अटैच्ड दिखा रहे हैं. Could you get it?
Yes, ma'am.
हाँ, so you find one strand will have 5 dash free, so that's why now you call the ends as 5 dash end and the 3 dash end. तो एक ही स्ट्रैंड को लेंगे तो ऊपर 5 डैश एंड फ्री रहेगा, तो उसके अदर एंड पर 3 डैश एंड फ्री रहेगा. Followed? Everyone understood or not? And then here they are showing how T and A joining and how C is pairing with the guanine. So C is pairing with the guanine. You can see 3 hydrogen bonds. And in the case of thymine and adenine, 2 hydrogen bonds are very clearly visible, formed between the first nitrogen, that is the nitrogen at the first position, in both the nitrogenous bases. और वहां पर 6th कार्बन से बॉन्ड हो रहा है. आर यू गेटिंग द फिगर?
Yes,
अंडरस्टैंडिंग?
ma'am.
ठीक है. अब ये सारी चीज आपके 11th में तो कोई नहीं पूछने वाला है एग्जाम में, बट इट्स वेरी वेरी इंपॉर्टेंट फॉर योर कॉम्पिटिटिव एग्जाम्स चिल्ड्रन. इज इट क्लियर? तो कॉम्पिटिटिव एग्जाम पॉइंट ऑफ व्यू से आप अगर पढ़ते हो तो whatever figures I have sent children, try to prepare them. Okay, but for your exam, if I ask you the question, so my questions will be, that is, number 1, describe the Watson and Crick model of DNA. If it is asked, you will draw only the first figure, that is the DNA double helix, which is helically coiled along with the ladder-like structure which is made diagrammatic representation. Did you get that diagram? तो वो दो diagram बना देंगे. Now my next short questions will be, write the differences between nucleoside and nucleotide. You can note down these questions. Okay, write the differences between nucleoside and nucleotide. Then my next question, what is the difference between deoxyribose sugar and ribose sugar? Explain with structure. Explain with their structural formula. Did you get it? All are noting the questions or not? Yes or no?
Yes, ma'am.
Okay. Okay. So how many questions you have got now?
3 questions.
Three questions. Now my fourth question will be that what are the nitrogenous bases? Okay, what are the nitrogenous bases? How are they classified? So you will say what are they? I have already sent those pages. If you read it, very nicely it's given. और जो अभी बादी has said that only will be written. That they are of two types, purines and pyrimidines. Then you will tell about their structure. Okay, and one diagram is given where pyrimidine ring and purine ring is made along with the numbers. Okay, so first position the nitrogen is there, फिर सेकंड में कार्बन है, थर्ड में फिर नाइट्रोजन है, फोर्थ कार्बन है, फिफ्थ कार्बन है, सिक्स कार्बन है. Okay, so the six membered ring is made up of how many nitrogen and how many carbon? Rumisha, do you have that diagram?
Yes, ma'am.
Can you tell me how many carbon and how many nitrogen make up that pyrimidine ring?
Ma'am, 2 nitrogen and 4 carbon.
Four carbon. ठीक है, nitrogen they are present at what position?
Ma'am, 1 and 3.
One and three. Okay, so for that reason only I wanted children that all should have the diagram in the notebook जिसे आप तुरंत देख के आप समझ सके. Now can you tell Neha in the purine ring, okay, to which carbon the imidazole ring is attached? Neha, do you have this diagram?
Yes, ma'am.
So can you tell me?
Ma'am, fifth carbon,
हाँ
fifth carbon,
हाँ
and
एक
the fourth one.
फिफ्थ एंड फोर्थ बिल्कुल ठीक है. तो डायग्राम सामने है आपके, तो आपको समझ में आता है कि फिफ्थ और फोर्थ कार्बन से now the imidazole ring is having how many carbon, how many nitrogen? It is a five membered ring. So
Three carbon and two nitrogen.
two nitrogen. Understood? So Barbie, do you also have these diagrams? These structures you have made of purine and pyrimidine? This is a general
Yes,
structure.
ma'am.
डन ना. हाँ, so these two are the general structures. फिर उसके बाद आपने specific structure you have made of adenine, then guanine, cytosine, thymine and uracil. Made everyone? Done na? Okay.
Yes, ma'am.
Now this was all about the DNA structure, what you have to study, children. Now I will be showing you one video and then I will start with the RNA. Okay, the functions of, so I have given three questions. उसके अलावा और क्वेश्चन क्या होता है, that is why do we say or why the two strands of the DNA molecule anti-parallel? Okay, that is very important question. Why the two strands are anti-parallel? Okay, now other question will be one more question which is asked in the exam, that is to which molecule the steps are bonded or the steps are connected, and to which carbon atoms. Which carbon atoms? Then which type of the bond is formed between the deoxyribose sugar and the phosphate? So what is that bond? Can you tell me what is that bond? Just turn the pages if you have noted. So last period the slide was showing the bond formed between the phosphate and the deoxyribose sugar.
Phosphodiester bond.
Phosphodiester bond. But tell me the carbon numbers also. What numbers we say?
5,
5 dash 5 dash 3
Huh?
dash.
5-3- phosphodiester bond. ठीक है. Okay, so that was all. Now I am presenting and try to see once again and understand it. The video you see. So, Neha, can you tell me why the two strands are anti-parallel?
Ma'am, I don't know.
Don't know. If you see that structure now, which one we have done, so then that is your 5.8 figure, children. So you will be able to answer. You have to explain in this. Just 1 minute. See this video also, and then
Ma'am, you are not clearly audible.
okay. Voice going. Screen is visible, children.
Yes, ma'am.
Okay. Am I audible now?
Yes, ma'am.
Okay. Now see the video.
Structure of DNA. DNA, deoxyribonucleic acid, is present in the nucleus of every cell. It is present as a highly coiled structure in the form of a chromosome. In 1953, James Watson and Francis Crick proposed the structure of DNA. DNA is a macromolecule polymer made of subunits called nucleotides. The nucleotides are made of 3 units: a phosphate group, a 5-carbon sugar called deoxyribose, and 1 nitrogen-containing base. The nucleotides are arranged into chains or strands. Each strand has a backbone made of alternating groups of deoxyribose sugar and phosphate groups. The sugar molecule of one nucleotide is linked with the phosphate of the next nucleotide by phosphodiester bond. There are 4 nitrogenous bases in DNA: adenine and guanine, called purines, and cytosine and thymine, called pyrimidines. The bases are attached to the sugar molecule. 2 strands of nucleotides pair together in a specific way. Adenine pairs with thymine, and guanine pairs with cytosine. 3 hydrogen bonds are present between C and G, while 2 hydrogen bonds are present between A and T. AT and GC base pairs are said to be complementary. A DNA strand is assigned a direction based on what group is found at the end of the strands. If one end has a free phosphate attached to the 5th carbon of the sugar, then that end is called the 5' end or 5th' end. If the other end of the strand has a free hydroxyl group attached to the 3rd carbon of the sugar, then it is the 3' end or 3rd' end. The 2 strands of DNA are anti-parallel to one another. One strand is 5' to 3', and the other is 3' to 5'. The paired DNA strands form a double helix, which looks like a twisted ladder. DNA contains the instructions needed for an organism to develop, survive, and reproduce. Genes carry biological information that is copied and transmitted to the next generation. Each time a cell divides to form two daughter cells. A stretch of DNA or DNA sequence contains instructions to make a protein and is known as a gene. In humans, the size of a gene varies greatly between 1,000 and 1,000,000 bases. Summary. Inside each cell lies a nucleus that contains long strands of deoxyribonucleic acid. The double helix structure of DNA is made of phosphate, deoxyribose sugar, and 4 nitrogenous bases. The strands of DNA are antiparallel in arrangement. DNA encodes genetic information.
Did you understand, children? Romisha?
Yes, ma'am.
Uh, to see this video once again. एंटी पैरेलल it is shown very nicely. अभी आपने जब वीडियो देखा तो you must be seeing that if in one strand A is written means corrected, in the other one the T is written inverted. ऐसा देखा था कि नहीं देखा था, इस पर ध्यान दिया? G
Ma'am,
सीधा लिखा है.
ma'am,
हाँ,
please you play again the video. I lost the connection.
okay. I'll do it once again.
Structure of DNA.
Now are you there, Rumisha?
DNA, deoxyribonucleic acid, is present in the nucleus of every cell. It is present as a highly coiled structure in the form of a chromosome. In 1953, James Watson and Francis Crick proposed the structure of DNA. DNA is a macromolecule polymer made of subunits called nucleotides. The nucleotides are made of 3 units: a phosphate group, a 5-carbon sugar called deoxyribose, and 1 nitrogen-containing base. The nucleotides are arranged into chains or strands. Each strand has a backbone made of alternating groups of deoxyribose sugar and phosphate groups. The sugar molecule of one nucleotide is linked with the phosphate of the next nucleotide by phosphodiester bond. There are 4 nitrogenous bases in DNA: adenine and guanine called purines, and cytosine and thymine called pyrimidines. The bases are attached to the sugar molecule. 2 strands of nucleotides pair together in a specific way. Adenine pairs with thymine, and guanine pairs with cytosine. 3 hydrogen bonds are present between C and G, while 2 hydrogen bonds are present between A and T. AT and GC base pairs are said to be complementary. A DNA strand is assigned a direction based on what group is found at the end of the strands. If one end has a free phosphate attached to the 5th carbon of the sugar, then that end is called the 5' end or 5th prime end. If the other end of the strand has a free hydroxyl group attached to the 3rd carbon of the sugar, then it is the 3' end or 3' end. The 2 strands of DNA are antiparallel to one another. One strand is 5' to 3', and the other is 3' to 5'. The paired DNA strands form a double helix, which looks like a twisted ladder. DNA contains the instructions needed for an organism to develop, survive, and reproduce. Genes carry biological biological information that is copied and transmitted to the next generation. Each time a cell divides to form two daughter cells. A stretch of DNA or DNA sequence contains instructions to make a protein and is known as a gene. In humans, the size of a gene varies greatly between 1,000 and 1,000,000 bases. Summary. Inside each cell lies a nucleus that contains long strands of deoxyribonucleic acid. The double helix structure of DNA is made of phosphate, deoxyribose sugar, and 4 nitrogenous bases. The strands of DNA are antiparallel in arrangement. DNA encodes genetic information.
So, how many students have understood? Raj, did you understand? Raj, are you there?
Yes, ma'am.
Yes, ma'am.
तो ये देख के कुछ समझ में आया? आप अब बोल सकते हो क्या? Can you explain the structure of the DNA now?
No,
Raj,
ma'am.
no. Why?
हम समझ लिया है पर बोल नहीं सकता.
क्या समझ आ गया है? तो क्या लिख सकते हो?
Yes, ma'am.
आप लिख सकते हो. ठीक है. Neha, when did we start this DNA structure? Can you tell me the date?
Ma'am, just a minute, I'm checking it.
Just turn the page and tell me which day we started with the DNA.
Ma'am, I think it's 28.
28 we started. अब आज फर्स्ट है तो 3 दिन हो गए आपको, 4 दिन, isn't it? So by now everyone should have all the figures plus all should learn it also. राज, आपको बोलने की भी हैबिट डालनी है. Is it clear? You have to express yourself in very simple language what we are doing. Okay, there is no need of mugging up the things which are in the textbook. Just see the diagram, children, and what is written, what is visible in the diagram. Try to say it in your own words. Followed all of you all? So how many have understood? Neha can express nicely now.
Yes, ma'am.
Okay, and two strands anti-parallel, you will be able to write down that in a DNA molecule, एक बार समझ लो अच्छे से, in a DNA molecule, we find that if one strand is having the, or it is having the 5 dash end on top and the other end is 3 dash, so in the other strand you will find it will have the 3 dash on top and the 5 dash end at the other end. So that's why we say it is anti-parallel. Followed?
Yes, ma'am.
Okay. So try to see it. Further you read those pages, try to see the diagrams once again, and then you have to prepare this DNA structure very nicely, children. Okay, and it's very, very important for your other exams also. Followed? Okay.
Yes,
Now
ma'am.
I will not start with the, sorry, I don't want cellulose. Now try to see, we have done the carbohydrates, proteins and fats. So RNA I am not starting. Few videos you see so that your proteins, carbohydrates, fats will also be revised. Okay, and try to cover up the whole DNA, and next period I will start with the RNA. So RNA is also a complicated structure. Here so we did only one type. Lekin RNA they are of 3 types, and all the 3 types you have to study in detail. Okay, and why is it important to study in detail? Because next year all that will be needed. Okay, so now try to see this video and then
Glycogen and starch. Polysaccharides are large biomolecules consisting of monosaccharides bonded by glycosidic bonds, also known as glycans. They can be one of three common types: cellulose, starch, or glycogen. In this session, we'll concentrate on glycogen and starch. Glycogen is a major carbohydrate reserve found in animals. Structurally, it is a branched chain polysaccharide made of alpha D glucose. The carbon atom number 1 from a glucose molecule is bonded to the fourth carbon atom of the next glucose molecule, forming an alpha 1,4 glycosidic bond. After every 8 to 10 glucose units of the linear chain, a new branch of the glucose chain will be produced. On the new branch, the C1 branching point of a glucose molecule will bond to the sixth carbon of glucose on the main chain. This will form an alpha 1,6 glycosidic bond. Glycogen functions as the energy storage form in both animals and fungi. In humans, it is stored mainly in skeletal muscle and the liver. Muscle glycogen breaks down into glucose for energy production, while liver glycogen breaks down into glucose for the maintenance of blood glucose levels. Starch is a carbohydrate reserve in plants, found as granules in tubers such as potatoes and tapioca, and seeds such as corn, rice, and wheat. It consists of two types of molecules: a linear and helical amylose and a branched amylopectin. In amylose molecules, alpha D glucose units are linked by 1,4 glycosidic bonds with the hydrogen bonding oxygen atoms O2 and O6 towards the outer side of the helix and the ring oxygen pointing inwards. Amylopectin is a branched structure with 1,6 glycosidic bonds at the branch points. Structurally, it is similar to glycogen but has fewer branches. In its pure form, starch is a white powder. Humans consume starch-rich foods for energy over a sustained period of time. Summary: polysaccharides are large biomolecules consisting of monosaccharides bonded by glycosidic bonds. Glycogen is a major carbohydrate reserve in animals. Glycogen is a branched chain polysaccharide made of alpha D glucose. Starch is a carbohydrate reserve found in plants. Starch consists of two types of molecules: a linear and helical amylose and a branched amylopectin.
So one more protein structure is also there that I'll show you next period, children, so that you can understand further better. So now we'll end up the class. I'm stopping the sharing. Ritu is saying 8th September. What 8th September we started? Okay, so now we'll do Shanti part. Try to revise everything, children. And if you still have the doubts, so then ask next period. And next period, we will start with the RNA. And what type of questions are asked, that also I have told you. So try to prepare that way, along with all the structural formulas of the different components of this DNA molecule. Okay, and send the PDF of all the diagrams of this DNA by tomorrow evening. Clear to everyone? You can write in the chat box. By tomorrow evening, I want all the figures of the DNA. Okay, which I have sent all the structures. You have to make it and send it. Okay. So now we'll do Shanti part, children. Om Shanti Shanti Shanti. Sai Ram, children.
Thank you, ma'am.
Sai Ram. You can leave,
Thank you,
children.
ma'am.
Thank you, ma'am.
Okay. Sai Ram.
Thank
Study,
you,
children.
ma'am.
Don't leave the